Good afternoon, everyone, and welcome to our 15th edition of our INFOGEST Viva webinar. My name is Hernandez, and I would like to introduce Shannon Gwala and also Cynthia Fontes, who recently joined the team, and both will conduct the webinar with me. This event is organized by a team of postdocs from Teagasc Food Research Centre in Ireland. This webinar aims to share the latest findings in digestion with presentation from the most recently graduated PhDs, postdoc, and young researchers. This webinar is being broadcasted live on YouTube and will be accessible on the INFOGEST YouTube channel. The presentation this time it's in a pre-recorded video. So will be followed by a question and answer session with the speaker. So I actively invite you to use the Q&A button that should be around here. Our speaker today is Ana Blanco Doval with a study of protein digestion of mare milk from different management system and during lactation. Anna is a postdoctoral researcher at Agroscope Research Center in Bern, Switzerland. She recently obtained her PhD in food science at the university in the Basque Country in Spain. There, she performed a thorough characterization of mare milk chemical composition, as well as a study of digestion of mare milk proteins at different lactation stages and belonging to different management systems. So thanks, Anna, for being here, and then the floor is yours.
Good afternoon. My name is Ana Blanco Doval. I am a postdoctoral researcher at Agroscope Research Center in Switzerland. I very recently defended my PhD thesis at the University of the Basque Country in northern Spain. In that PhD thesis, I characterized the chemical composition of mare milk from an autochthonous horse breed from the Basque Country, which is Basque Mountain horse. And within that research, we studied the digestion of mare milk proteins. So that's the part I'm going to talk about today. And I entitled this presentation, The Study of Protein Digestion of Mare Milk from Different Management Systems and During Lactation. I'm going to do a very brief introduction. So, mare milk is mainly consumed in Central Asia, in countries like Kazakhstan, Kyrgyzstan, Uzbekistan, Belarus, Mongolia, etc. In these countries, mare milk is a traditional food that is mainly consumed in its fermented version, which is called kumis. And in these Asian countries, mare milk has widely been used as a medicine or as a therapeutic food to treat several diseases like tuberculosis, chronic hepatitis, peptic ulcers, heartburn, as well as to help patients expectorate, facilitate digestion, lower fever, stimulate the nervous system, boost strength, etc. I brought an example of this here in this picture. This is the Lesnoye Sanatorium in Russia. In this sanatorium, they treated patients with tuberculosis using kumis. Moving to Europe, mare milk consumption is mainly concentrated in Ukraine, Hungary, Austria, Bulgaria, and Germany. And in these countries, mare milk has widely been used as an alternative to human breast milk due to two main reasons. The first one is that mare and human milk are very similar in composition. And the second one is that mare milk has presented low allergenicity in people with cow milk protein allergy, which is an allergy very common among children younger than 3 years old. However, in these European countries, the trust on the therapeutic properties of mare milk still remains. Mare milk is mainly marketed either as an ingredient in cosmetics or as a food itself, in a liquid form, in a powder form, or as nutraceuticals. And both producers and marketers very often claim those beneficial properties of mare milk. However, there's very limited research that supports these beneficial properties. There's limited research even regarding mare milk composition, particularly those minor compounds or those bioactive compounds. But there's also limited research regarding the allergenic response of mare milk, the functional properties of mare milk, as well as regarding the digestion of different nutrients in mare milk. However, we do know some things about mare milk. So mare milk is very poor in fat or has a very low fat content. Protein content ranges between 1 and 2%, and lactose content ranges between 6 and 7%. If we compare these values with milk from other mammal species, we can see how mare milk is actually very poor in fat, and this is more similar to human milk, which is also poorer in fat compared to milk from ruminant species. In the case of proteins, mare and human milk are very similar in protein content, and overall they have a lower content than milk from ruminant species. And the opposite occurs in the lactose content. In this case, mare and human milk present higher contents of lactose compared to milk from ruminant species. Regarding the proteins in milk, in ruminant milk, about 80% of milk proteins are caseins and only 20% are whey proteins. However, in mare milk, about 55% of milk proteins are caseins and the rest are whey proteins. So, compared to ruminant milk, mare milk contains more whey proteins and a lower amount of caseins. And this is actually more similar to human milk, which contains about 70% of whey proteins and only 30% of caseins. So, mare and human milk are considered milks rich in whey proteins, whereas ruminant milk is usually considered milk rich in caseins. And mare and human milk also present higher amounts or higher contents of immunoglobulins and non-protein nitrogen compared to ruminant milks. Um, in the case of protein digestion, we only found two significant studies that addressed digestion of mare milk proteins. The first one is a study from 2010. In this study, they compared protein digestion in milk from different species. They used human gastric and intestinal fluids, and they performed a two-step in vitro digestion of 30 minutes each. The second study was one from 2023. In this case, they also compared digestion in milk from different species, and they used an infant in vitro digestion simulation. If we observe the main results obtained in both studies, we can see how both studies observed a high casein degradation during the digestion. However, the first study found that alpha-lactalbumin from mare milk was quite resistant to both gastric and intestinal digestion, and that beta-lactoglobulin was highly degraded during intestinal digestion. On the other hand, the second study observed how both beta-lactoglobulin and alpha-lactalbumin are resistant to the gastric digestion, but they are both degraded during intestinal digestion. In both studies, they did not differentiate between the two beta-lactoglobulin isoforms that are present in mare milk. So, the aim of our study was first to determine the digestibility of mare milk proteins during simulated in vitro human gastrointestinal digestion using the extended INFOGEST protocol. We also aimed to describe the peptide release during gastrointestinal digestion of mare milk, as well as to identify potential bioactive peptides that could be released during digestion. And finally, we aimed to assess the effects of management system and lactation stage on the studied parameters. So these were the materials and methods we used in this research. Mare milk samples were obtained from 3 commercial farms located in the region of Araba in northern Spain, particularly in these 2 zones of the region. Each commercial farm milked 6 individual mares during the complete lactation period, which was from May to October, and we obtained a total of 311 individual mare milk samples. The main difference among farms was the feeding regime of the mares. So in farm 1, mares were kept on pasture or under grazing conditions only during May, and afterwards they were fed a mixture of alfalfa, silage, hay, fruits, and potatoes. On the other hand, mares from farms 2 and 3 were kept on pasture during the entire lactation period, and only after July, when grass availability was scarce, Farm 2 supplemented mares with hay, and Farm 3 supplemented mares with silage. So in general terms, the main difference among farms was the length of the grazing period. It was shorter in Farm 1 and longer in Farms 2 and 3. All those mare milk samples were analyzed to characterize the chemical composition of milk. So this characterization included gross composition, mineral elements, fat and water-soluble vitamins, fatty acids, polyphenols, and antioxidant capacity. And then we wanted to focus on the protein fraction, and for that we characterized the whole proteome of mare milk, and we studied protein digestion. And this is the part of the research that I'm going to talk about today. So, digestion of, or the study of protein digestion was done in collaboration with Agroscope Research Center in Switzerland. For this, we used the INFOGEST protocol for protein digestion, which includes 3 different phases. The oral phase, in this case without amylase, the gastric phase with pepsin, and the intestinal phase with bile and pancreatine. From the 311 mare milk samples, we selected 54 samples for this study, and we approached a mare milk protein digestion study from 2 perspectives. So in the first one, we assessed total digestibility of proteins. For this, we subjected milk samples, to intestinal digestion, which means the complete digestion process from oral, gastric to intestinal digestion, and both the digestible and the indigestible fraction were analyzed for total amino groups using the OPA method, and for for individual amino acids using liquid chromatography. Individual amino acids from raw mare milk were also analyzed, and we also analyzed total nitrogen in raw milk using the Geldof method. And then we characterized the peptides that were released during digestion of mare milk. In this case, each sample was digested twice. The first digestion was stopped after the gastric phase, and the second digestion was a complete digestion, which included oral, gastric, and intestinal phases. And digesta were analyzed using liquid chromatography coupled to a linear ion trap to identify the peptides released. We also performed gel electrophoresis in order to identify the main proteins present in mare milk, and we compared the peptides released only with those major proteins. And finally, we subjected all data to statistical analysis. So on one hand, we performed or we used the general linear model of the analysis of the variance to study the effect of farm or management system, lactation stage, and the interaction effect between farm and lactation stage. Then we subjected data to a stepwise discriminant analysis in order to have a multivariate perspective of the results. Also to classify samples according to management system or lactation stage. So now I'm going to expose the results we obtained. And we observed that mare milk proteins had a digestibility of 94 or 96% depending on the analytical method used, and that most of the or all amino acids presented a digestibility higher than 90%, except for alanine, isoleucine, serine, and threonine. Okay. This graph represents the DR values obtained for infants, children, and adults. And as we can see, the limiting amino acid in infants was threonine, and the limiting amino acid in children and adults was histidine. To study the release of peptides, as I said, we first identified the major proteins in mare milk using gel electrophoresis, and we identified 4 caseins, including alpha S1, alpha S2, beta, and kappa casein, and 4 whey proteins, which were beta-lactalbumin 1 and 2, alpha-lactalbumin, and lysozyme C. We studied changes in the size of the peptides liberated after gastric and intestinal digestion, and we observed that the size of the peptides liberated from all caseins, beta-lactalbumin 1 and alpha-lactalbumin A, significantly decreased from gastric to intestinal digestion. Conversely, the size of peptides liberated from beta-lactoglobulin 2 and lysozyme C did not significantly differ from gastric to intestinal digestion. So this suggests that there's some kind of resistance of these 2 proteins to at least intestinal digestion. We confirmed those results with the heat maps we built with the abundance of peptides liberated after gastric digestion in the upper part of the graphs and after intestinal digestion in the lower part of the graphs. So as we can see here, all caseins in mare milk were highly degraded already during gastric digestion, and they were further degraded during the intestinal digestion. In the case of whey proteins, both beta-lactoglobulin 1 and alpha-lactalbumin A were already degraded during the gastric digestion, and they were also further degraded during the intestinal digestion. But on the contrary, we did not find many peptides liberated from beta-lactoglobulin 2 and lysozyme Z, even during gastric digestion. So this again suggests that these 2 proteins present some kind of resistance to gastric and intestinal digestion. Then we searched for bioactive peptides liberated after gastric or intestinal digestion, and although we did not find any bioactive peptide per se, we did identify a number of precursors of this bioactive peptide marked in green here. This is a peptide located between the amino acids 191 and 200 within the amino acid sequence of beta-casein of mare milk. And this peptide has been described to be an inhibitor of the angiotensin converting enzyme, so it might have some antihypertensive functionality. With all the data obtained from protein digestion, we did a statistical analysis to study the effect of maintenance system and lactation stage, and we did not find very significant effects overall. We did find some DR values that were significantly affected by any of the factors studied, but these significant differences came from differences in the amino acid profile of raw milk and not from differences in digestibility of individual amino acids. We also applied the statistical analysis to the abundance of peptides liberated after gastric or intestinal digestion, And as we can see, there are not many peptides that are significantly affected by any of the factors studied. And the majority of those peptides that were affected by management system. Very few were affected by lactation stage. In addition, most of these peptides were liberated after gastric digestion, and only a few peptides liberated after intestinal digestion presented significant differences according to any of the factors studied. And this was something that we expected, considering that those peptides liberated after gastric digestion are further degraded during intestinal digestion. And finally, we applied a multivariate analysis to all data in order to classify peptides or values obtained from the analysis after gastric and intestinal digestion according to the management system or the lactation stage. So if we focus on the effect of management system, we can see how the classification was not great, but the model was able to differentiate samples from Farm 1, which were those from mares with a shorter grazing period, from samples from mares that grazed for a longer period of time. And in the case of lactation stage, the model was able to classify mainly samples or differentiate mainly samples between late and early lactation stages. We can see that here as well. However, the best classification was obtained from data after gastric digestion and considering the effect of lactation stage. So most or highest differences in this sense were obtained or were differences between early and late lactation. So which were the conclusions of this study? Well, first, that mare milk is one of the most digestible dietary protein sources, that beta-lactoglobulin 1 and 2 isoforms perform differently during digestion, that beta-lactoglobulin 2 and lysozyme Z present resistance to digestion, alpha-lactalbumin A degrades during digestion, and that lactation stage and management system significantly affect some of the parameters related to protein digestion, and therefore behavior of mare milk proteins during gastrointestinal digestion could be modulated through diet or optimized by selecting milk from specific lactation stages. And as a general conclusion, further research remains essential to elucidate the role that human digestion has on degradation and functionality of mare milk proteins. And just to finish, all this work resulted in a publication that has been very recently published, so you can access it and get more detailed information about the work we did. So this was all. Thank you very much for your attention.
Okay. Thank you so much, Anna, for your interesting presentation, and congratulations for your recently defend of your Viva. So now we are gonna start with the Q&A section. I think we have some questions in the chat. So the first one is, what was the criteria for selecting the 54 samples out of more than 300 samples?
So we selected samples from half of the mares, so 3 mares per farm, and from only one lactation point each month. So in total, it was 6 lactation points or 6 time points during lactation, 3 mares and 3 farms.
Okay, great, thank you.
Within the peptide bioactivity study, you mentioned that no peptides with known bioactivity were found. To which compounds present in milk do you think the therapeutic benefits are attributed?
It can be due to a variety of compounds. Some of them attribute those beneficial properties to the lipid fraction, so to some unsaturated fatty acids. Some talk about a high quantity of lysozyme or lactoferrin, and some others could be, for example, in my PhD, we also measured the antioxidant activity, and it was not very different to some other milks. So there can be a variety of compounds that take part in that bioactivity or that functionality of the product. But as I said, it is not very widely studied for the moment. So at this moment, we cannot talk about a specific bioactive compound having or being responsible for the functionality of the product. So first, we need to deeply characterize all the compounds in the product and do more research on this area.
Thank you.
I think we have another question here. What's your opinion on the generation of this kind of results for the nutritional needs for infants when the in vitro digestion model it was applied for adults? What do you think?
Um, So I think that for sure we would need to replicate this study using an infant simulated in vitro digestion, because results are not completely replicable. In this sense, it would be very interesting to study, for example, the degradation of some of the proteins that are most responsible for the allergenicity of milk, because it is used for infant nutrition, mainly because it is supposed to be low allergenic. So yeah, of course, we would need to do more studies using infant simulations or even some clinical studies to see if those allergenic proteins are really degraded during digestion, and also to study if any other bioactive compounds are produced using the infant digestion. And yeah, we would need to replicate the study for sure.
Okay, thank you.
We have another question related with the allergenicity. Why do you think is Mars milk less allergenic?
Mm, I'm not really, I mean, it has not been very well described. So, of course, the casein, the lower amount of caseins can play a role on that, but we also find beta-lactoglobulin, for example, which is also allergenic. So, actually, the reason why it is said that mare milk is presents lower allergenicity is because two clinical studies that were done. So the research we have on that is not robust enough to really claim that mare milk is not or presents a lower allergenic response to people with cow milk protein allergy. So we still don't really know if that's actually true, and if it was true, why is it?
Okay. And the last question, I think, why does the farm have an effect on the peptide release? It is related to the type of feeding?
So it is probably related with the composition of mare milk. So in my PhD, in the other studies I did on different compounds in mineral elements or fatty acids, we observed that different farms produce a milk with a different composition. Sometimes it was significantly different. So probably a different composition of fatty acids or minerals can affect the digestion of proteins in milk. So differences are probably due to differences in composition.
Okay, thank you, Anna.
I think that we are done with the all the questions. So thank you, Anna, for your interesting discussion as well and the presentation. So now I'm gonna share my screen. So Shannon, it's gonna advertise.
Yeah, thank you, Anna, for your concise presentation there. And I will just announce our next webinar, which will be held on the 2nd of October. And we are going to be having Charlotte Oliver, who's going to be speaking on the in vitro assessment of milk extracellular vesicles, its digestion and bioactivity. So please tune in again next webinar on the 2nd of October, same time. We will send out a registration link so that you can all register and be notified of our webinars in due course. Our next announcement is on the INFOGEST video abstracts, which was announced during the INFOGEST conference in Porto, and we are calling people to submit their expression of interest, where they can make a 3-minute video based on a poster presentation or based on a recently published paper. This video will be posted on LinkedIn, and in that way, you can disseminate your newly published paper to a wider audience. I think with that, we can call it a day. Thank you for tuning in and see you next time.
See you. Thank you.
Thank you. Bye.